Hello everyone and welcome to Micro Mahasutra. This is our General Microbiology class 9th. We will be discussing general bacteriology and the topic for today's discussion is culture media and culture methods. Okay. So culture media and culture methods are the core topic of microbiology and this is really important for any examination. Let us now discuss about the different culture medias. First of all, let us understand what is enriched media. For example, some people drink normal milk. Okay. Some people add Bonvita and Horlicks in it. Why? To enrich the media. So if you are a normal person, you will grow on milk. But if you are a fastidious person, fastidious means which don't like the ordinary things, you will grow on some enriched media. Now you must have thought ki, am I fastidious or non? So let me know in the comment section. Okay. So basically enriched media means the ones having basal media just like the normal milk plus some extra growth factors, that is called as enriched media for the fastidious organisms like blood agar. So normally agar is there but blood is responsible for the growth factor. Serum agar, serum is the growth. Egg agar, egg is the growth. Loeffler's serum slope. So serum is again for the growth. So remember we have told enriched media, not the enrichment. Okay. What is enrichment? Enrichment means it promotes the growth of desired organism and inhibits the others. So the one which promotes the growth of one organism and inhibits the growth of other is not enriched, it is enrichment. It is enrichment, and the good part is that it is always liquid. Enrichment medias are always liquid. So you will have it alkaline peptone water in the example or tetrathionate broth. What is broth? Broth is also liquid. Okay. So tetrathionate broth and alkaline peptone water. You know that Vibrio loves to grow in alkaline pH. So Vibrio is an example of alkaline peptone water as the culture media. Then broth. So tetrathionate, what does it mean? Thio means sulfur and S for sulfur, S for salmonella. Tetrathionate has word thio means sulfur and this sulfur has salmonella, S for S. Coming on to selective media, remember all the selective media, S for selective, S for solid, all of them are solid. Let's have an example. For example, PTA media. So PTA media, potassium tellurite agar. Potassium tellurite agar is for diphtheria. Okay, diphtheria. You got it? Yes. TCBS agar. TCBS has C, so Vibrio cholera. LJ media, Mycobacterium. So LJ media for what? For Mycobacterium. And DCA and XLD media for Salmonella and Shigella. Salmonella and Shigella. Okay. Full forms we will discuss while discussing the one-on-one bacteria. But for now, just remember that if you have PTA, okay, PTA. So we used to say that dip the coffee in apt amount. So dip the coffee. Dip means diphtheria. Coffee, C for Corynebacterium diphtheria, in apt amount is APT. Okay, like that. But those of you who know Hindi very well, उसका पता करो जिसके पास granules हैं. The one who is having granules, take his address. Pata in Hindi is address in English. Okay. So granules, who is having the granules? The diphtheria is having the granules. So pata PTA. Okay. Vibrio cholera, C and TCBS, easy to remember. So here VC, Vibrio cholera, TCBS is going to be the agar. LJ media, LJ. So please remember Mycobacterium tuberculosis, okay, TB, okay. So remember T and L are very same, okay. So TB and LJ media like that. And The one which is starting with D, the one which is ending with D has got S and S. 𝗲𝗃, that is going to have Salmonella, Shigella. Coming on to differential media. Differential media means it differentiates between the characteristics of bacteria. Let me give you a beautiful example by this picture. Can you see? Can you see the pink material, the pink organisms? So which media is this? MacConkey media. So you have seen pink medias on MacConkey. So now this is a differential media. That means on MacConkey Agar, the lactose fermenters are pink, whereas non-lactose fermenters are colorless. On MacConkey, the lactose fermenters are pink and the non-lactose fermenters are colorless. Okay, right? And then you have got the clad media also. Clad media. Okay, clad media also differentiates. Okay, how to remember? You have got LEDs, right? So LEDs differentiates in darkness, and whenever you will switch on the LED, it will turn the room into brightness. So darkness, brightness, LED. So clad is also a differential media. Indicator, yes, indicating the fermentation, lactose fermenter. So indicates the reaction. Which agar? MacConkey Agar. MacConkey Agar, because it is indicating that some reaction is taking place. That is why we call it as indicator media. Coming on to transport media or holding media. See, my dear friends, in the time of COVID, we used to visit the people's home, and we used to collect the sample. Then we used to take it to our laboratories. In what? In the transport media. But we used to take care of two things. One, that the number of organisms should not multiply, because if they will multiply during the transport, obviously we will have a false reading, a wrong reading, that these many bacteria are present in that individual. You got it? That's why we have to keep a check that the transport media should prevent the multiplication, but it should maintain the viability. We have to make sure that the bacteria is viable, that the bacteria is not dead. Okay, you got it? Yes. And we have to transport. Now the mnemonics is PVR. So how I remember? Transport me, please somebody transport me out of these books to the PVR. What is PVR? P for Pikes Media, V for VR, Venkatraman Media, and S for Stewart's Media. Okay, PVRs. So P for Pikes, VR is VR, and S for Stewart's Media. How to remember? P for P, Streptococcus pyogenes is Pikes Media, VR media is Vibrio, right? And just the Gonococcus has got Stewart's Media. This you have to remember, that Gonococcus has got which media? They have got Stewart's Media. Okay, you got it? Good. Coming on to anaerobic media. So cooked meat. See, when the meat is not cooked, you put the meat on flame, and flame, fire. By 8th and 9th standard knowledge, you must be knowing that flame or fire requires oxygen. So all the oxygen is being carried by flame. So you are left with no oxygen. So these medias are devoid of oxygen. What I wanted to tell, that Robertson cooked meat media has no oxygen. Coming on to the next one, Bat's Agar. This is also having no oxygen. Okay. Thioglycolate broth. So thio means again sulfur but no oxygen. So thioglycolate, Bat's Agar, Robertson cooked meat media, example of anaerobic media. Fermentation media. So this is a media, you kept a glass on it, there will be gas release and the gas will be seen here. So this is a fermentation media. So this is a tube. Which tube? Durham's tube. Okay, this is a Durham's tube used to determine the fermentation, because fermentation has a release of gas. Coming on to culture method. So there are different culture methods. One of the culture method is streak culture. Okay, I'll change the color. Streak culture. Streak culture means we take a sample. For example, you took a sputum sample. Okay, you took a sputum sample and you put the sputum sample over here. Once you put the sputum sample over here, you make different streaks. And at the time of making different streaks, my dear friend, you can isolate the different bacteria. Some might be blue ones, some might be red ones, some might be green ones. So it is used to isolate the bacteria from a mixed population. Okay, coming on to the next one, lawn culture, carpet culture. Lawn culture or carpet culture means that you are equally spreading the substances, just like this, equally spreading the substances all around, right? So this is used to check the antibiotic susceptibility. So what you did, okay, I'll show you once again. What you did, you put a culture plate and you spread the material. Okay, this is the material which I am spreading. After spreading the material, bacteria started to grow. So this is the growth of bacteria which is happening on the material. And now that my bacteria has grown, what I did, I took an antibiotic and pasted it here. This is my antibiotic. For example, this is 𝗵𝗵𝗵𝗵 antibiotic. Okay. So after some time, what I saw that the bacteria surrounding this antibiotic started to disappear. The bacteria went away. So this way I can understand that 𝗵𝗵𝗵𝗵 can kill that bacteria. That means bacteria is susceptible to 𝗵𝗵𝗵𝗵. Okay. So please understand, this is the method, the lawn carpet or culture method is used to check the sensitivity of the antibiotics. This is also called as disc diffusion method because we put down a lot of discs. So this is a disc diffusion method, okay, for antibiogram. So which medium did you use? The medium used is Muller-Hinton Agar. Which media did we use? Muller-Hinton Agar. Culture media is Muller-Hinton Agar. Method is disc diffusion method. So Muller-Hinton hunts the antibiotics. So who hunts bacteria? Antibiotic. So which is the agar? Muller-Hinton Agar. Okay, like that. Then stab. So stab culture is also important. So what we do, we stab the organism into this culture. Okay, so again I'll show you like this, just a second. Yeah, so see, this is filled with the culture. What I did, I stabbed the bacteria. After some time I saw this was the growth pattern of bacteria. This is suggestive of that mostly organism is growing on the top and very less organism are going in the bottom. That means these organisms first of all are motile, that means they can walk from here to here, and the second part is that they love oxygen because they are growing on the topmost layer. Whereas some non-motile anaerobes are there who want to grow here. They don't want to go here. I mean, they can't grow. So they are non-motile and they are anaerobes also. Okay. Non-motile examples, Klebsiella and Staphylococcus, we have already discussed, not discussing again. Coming on to anaerobic culture method. So what media was there? Yes, I just told you anaerobic culture media. So anaerobic culture media is Robertson cooked meat media, thioglycolate broth, Bat's agar. But now I am going to discuss anaerobic culture methods, just like candle jar, because candle wants to light, and whenever there will be ignition, all the oxygen will be utilized by the candle. That's why it is an anaerobic media. Even the Macintosh filter, it has got crystal of substances that are scavengers of oxygen. So they have got some crystals of substances that are going to absorb all the oxygen, and there will be anaerobiosis which will be happening. Similar is the case with the gas packed jar. So these are some images. Can you see? This is the stab culture which they have shown you. These are different streaks, so streak culture, and this is uniform, so this is lawn culture. This is a photo of blood culture. This is a photo of chocolate color, chocolate agar, blood agar, chocolate agar. Actually what happens is blood agar after some time releases hemosiderin, okay, makes hemosiderin. That's why there is a chocolate color, so this is called as chocolate agar. This I have already told you, MacConkey, which is going to be pink if it is a fermenter, and it is going to be colorless if it is a non-fermenter. Then what is this green one? Green one is the TCBS Coming on to this is PTA, potassium tellurite agar. PTA, I have told you, uska pata karo, which has got storage. So storage granules are, who is having the storage granules? The storage granules are with the Corynebacterium diphtheria. And what is this? This is LJ media. I told you L and T looks very similar. So this is for the Mycobacterium tuberculosis. Okay. This one, yes, alpha hemolysis, beta hemolysis, gamma hemolysis. I have already discussed this when we were discussing what? When we were discussing the gram positive bacteria. Then I told you that there are 3 types of hemolysis which are there. Okay. So these 3 are the examples. Don't worry, we will discuss in detail, right, when we will discuss the streptococcus. What is this now? This is pink one and this is orange color tube. This is the urease media. I'll discuss when we will discuss the biochemical analysis. Then we will discuss about the urease medium also. Okay. So thank you so much. Just as the nutrients act as the growth factor for the bacteria to grow, I just hope that you also grow many folds day by day and become a very successful doctor. Thank you so much.